One of the most important roles of carbon dioxide in the body is in acid-base balance. As a result, I've put together 3 tutorials looking at carbon dioxide and acid base. The first one I call Acid Basics, that just explains the basics of acid base and why carbon dioxide is important. Fundamentals of Anesthesiology and Critical Care. With me, your host, Pat Nelligan. Understanding gases and vapors. This tutorial is on carbon dioxide and its acid-base effects. Part 1, Acid Basics. This is the first tutorial in a series on the acid-base effects of carbon dioxide. To understand this topic, we need to have a basic understanding of modern quantitative acid-base chemistry. The content and terminology may be unfamiliar to some of you, but if you follow these tutorials, you will have an infinitely greater understanding of acid-base than most of your colleagues. In the previous tutorial, we looked at carbon dioxide content and delivery, carbon dioxide metabolism, the diffusion characteristics of CO2, how carbon dioxide interacts with hemoglobin, the Bohr and Haldane effects, and indeed how chloride is pumped into the red cell and bicarbonate pumped out. That's the hamburger effect or chloride shift, and how CO2 falls rapidly with increased mechanical ventilation and rises slowly with reduced mechanical ventilation. Then we looked at hypercarbia and bicarbonate in acute and chronic states. In this tutorial, we're going to look at carbon dioxide and acid base. I'm going to start with the absolute basics of acid base, strong ions and carbon dioxide, how hemoglobin buffers carbon dioxide, bicarbonate and chloride, and hyperventilation and hypoventilation. Let's start with the basics. The body is made up of water. About 60% of your body weight is water distributed between the intracellular space and the extracellular space, but virtually none of that is liquid. It's a gelatinous matrix. All biological and chemical reactions in the body occur in water, and dissolved in that water are gases like oxygen, carbon dioxide, and nitrogen, uncharged particles like glucose, urea, amino acids, and fats, and then charged particles. They're cations and anions, positively charged and negatively charged particles that may be mineral electrolytes or organic compounds such as proteins. Amongst the organic anions, we have lactate, ketones, Krebs cycle, metabolites, urate, oxalate, sulfate, etc. Water itself dissociates into its component parts. A moiety that contains a positive charge, we call them hydrogen ions, and a moiety that contains a negative charge, we call them hydroxyl ions, but they don't exist independently. At 37 degrees Celsius, the activity of both is about 160 nanoequivalents per liter. That is chemically neutral, and the pH, the negative logarithm of that quantity, is 6.8. All hydrogen and hydroxyl moieties in the body are associated with water dissociation, and water has a dissociation constant that is impacted by temperature. There are no free protons anywhere in the body that can be measured because they are clumps of water with a positive charge. But for the purposes of discussing acid base, I'm going to call them hydrogen ions. At 37 degrees Celsius, arterial plasma has a hydrogen ion activity or concentration of 40 nanoequivalents per liter or 40 nanomoles per liter, and that is vanishingly small. I'll talk about that again in a minute. Because it is such a small number, this is converted using a negative logarithmic scale into pH. You're turning very small numbers into a large number. You are laundering the number for all intents and purposes. The normal pH of arterial plasma is 7.4, and the pH of venous plasma is 7.37. This graph demonstrates the relationship between the pH scale on the x-axis and the hydrogen ion activity on the y-axis. And neutral pH is 7. That's when the quantity of hydrogen ions and the quantity of hydroxyl ions are equal. But in extracellular fluid, in arterial plasma, the pH is 7.4. At that level, there are 40 nanomoles per liter of measurable hydrogen ion activity. To put into perspective how small these numbers are, let's look at India in the year 2000. In the year 2000, India had a population of 1 billion. 1 nanomole is 1 billionth of a mole. If the red dots represent hydrogen ion equivalents, in the space of 1 billion, there are only 40 red dots. In comparison, the amount of sodium that is present in the same plasma is 140 millimoles. That is 140 million red dots. It represents the population of Uttar Pradesh in India, the same as the population of the UK, France, and Ireland. The number of sodium ions compared to the number of hydrogen ion equivalents is staggering. We overvalue the hydrogen ion activity because it is biologically important and because we are a little misled by the pH scale. But even in the extremes in biology, the number of hydrogen ions that would be calculable or measurable would be vanishingly small. Changes in the relative ratios of charged particles impact water dissociation equilibrium. When there is a relative increase in anions present relative to cations, then the quantity of positively charged water molecules, that's hydrogen ions, increases, leading to acidosis. When there is a relative increase in cations relative to anions, then the quantity of positively charged water molecules, hydrogen ions, decreases, and we have alkalosis. All of the positive charges in the body must equal all of the negative charges. Here are listed all of the charges in extracellular fluid, cations on the left and anions on the right. The total charge carried by cations is about 145 milliequivalents per liter, and there are an equal number of anionic charges. And note that I'm talking about valency here, not quantity, so it's milliequivalents, not millimoles, because some particles carry more than one charge: magnesium, calcium, and phosphorus. All of the positive charges are associated with hydroxyl ions, and each of the negative charges are associated with hydrogen ions. But those ions bind together to form water, such that at rest there is a slight excess of hydroxyl over hydrogen in extracellular fluid, hence the pH of 7.4, not 7.0. Each of the particles described functions differently in physiological conditions. The ones highlighted in white are known as strong ions. At all biological pH levels, they never bind to other particles. In other words, other ions. They don't form salts. The big players here are sodium, potassium, chloride, and organic particles such as lactate and ketones. Highlighted in yellow are the weak acid buffers. These particles or compounds are charged and contribute to electrical neutrality, but they are also capable of binding hydrogen ion equivalents and preventing a devastating fall in pH. The total electrical charge carried on strong cations is about 145 milliequivalents per liter. The charge carried by chloride is 105 milliequivalents per liter, and that leaves a difference of 40 milliequivalents per liter. That difference is known as the strong ion difference, or the SID. The majority of the difference in electrical charge between the strong anions and the strong cations, the SID, is made up of bicarbonate and albumin. Bicarbonate is about 24 milliequivalents per liter and albumin is 11 milliequivalents per liter. What is left over is a gap. You might know this as the anion gap, a gap in anionic charge, and it's made up of phosphate, lactate, ketones, and unmeasured anions. And that is usually about 5 milliequivalents per liter. Regardless of what charge is on each side of the equation, electrical neutrality must always hold. In any situation where the relative ratio of strong anions to strong cations increases, there is a reduction in the strong ion difference. This is the major cause of metabolic acidosis that we see in clinical practice. For example, it's caused by iatrogenic hyperchloremia from saline resuscitation. 0.9% saline contains sodium and chloride in a 1-to-1 ratio. Extracellular fluid contains sodium in a 1.4-to-1 ratio with chloride. As a consequence, when you give isotonic saline, the chloride level rises relative to sodium and the patient develops hyperchloremic acidosis. We also see this in ketoacidosis, lactic acidosis, and renal acidosis. And although a range of buffering mechanisms massively curtails hydrogen ion activity in extracellular fluid, the result is a fall in the pH. Conversely, when the total charge carried by strong cations exceeds the charge carried by strong anions, the strong ion difference increases, associated with a reduction in the hydrogen ion activity, a rise in the pH, and metabolic alkalosis. This process can occur due to sodium gain, such as during blood transfusion, sodium is retained and the citrate is metabolized. Remember, the blood is anticoagulated using sodium citrate. You can see this with chloride loss associated with vomiting or other GI losses, or loss of free water, that's contraction alkalosis. A fall in the serum albumin also causes metabolic alkalosis independent of SID. It is noteworthy that the pH of extracellular fluid is quite alkaline. When we do a blood gas, the pH of arterial blood is 7.4, and the reason for this appears to be teleological for safety. Alkalosis is relatively harmless, but acidosis negatively impacts transcellular ion pumps, neurological and cardiovascular function, etc., and the kidneys are programmed to keep the SID of major cations versus anions at 40 milliequivalents per liter. And most of that difference in electrochemical charge is balanced by bicarbonate. Most of the CO2 in the blood is bicarbonate, and this is an important component of homeostasis. Although additional production of CO2 generates both hydrogen ions and bicarbonate, generally the plasma pH and bicarbonate concentrations are kept stable by adjustments in alveolar ventilation. So, let's define acidosis and alkalosis. If the pH is less than 7.35 on a sample of arterial blood, then acidosis exists. The lower the pH, the greater the number of hydrogen ion equivalents, the worse the acidosis. Again, this is associated with all kinds of problems and disruptions to homeostasis, particularly with transcellular ion pumps, leading to vasoplegia, cardiac arrhythmias, and cardiac dysfunction. It is important to understand that the underlying pathology, for example, ketoacidosis or acute kidney injury, is a greater problem than the acidosis itself. Most of the devastating causes of acute critical illness cause acidosis. In general, for the same level of pH, metabolic acidosis is worse than respiratory acidosis because of the underlying problem. If the pH of an arterial blood sample is greater than 7.5, then alkalosis exists. Because this is on the flatter part of the pH curve, there are fewer associated medical conditions. By convention, we describe acid-base abnormalities as being either caused by fluid, electrolyte, and metabolic abnormalities. This is electrochemistry, and we talk about metabolic acidosis and metabolic alkalosis. We term disorders of CO2 control and elimination respiratory acidosis or alkalosis. Unique to the respiratory side, we divide respiratory acidosis into acute and chronic forms. Although both types of acid-base abnormality, metabolic and respiratory, are described independently, they massively overlap, as we shall see. Finally, to wrap up this section, I want to reiterate some information about quantities. Acids are produced constantly in millimolar quantities. These are organic acids like lactate, ketones, and various other anionic metabolic junk products. We also constantly produce carbon dioxide. These are all acids. Each millimole of anion or carbon dioxide will produce a millimole of hydrogen ions. That is a spectacular amount of hydrogen ions, but due to buffers, that quantity is reduced to nanomolar concentrations. So what are buffers? To prevent massive swings in pH and collapse of homeostasis, the body has developed a series of mechanisms to limit the fall in pH associated with metabolic activity. Intracellular and extracellular moieties can bind hydrogen and hydroxyl ions temporarily and stabilize the pH. These are known as buffers, and hemoglobin is the most important buffer of all. Carbon dioxide is an acid. Up to 90% of carbon dioxide produced is converted to carbonic acid that immediately dissociates to release hydrogen ions and bicarbonate. As net hydrogen ions are generated, the hydrogen ion concentration increases, lowering the pH. So CO2 is an acid, and importantly, this is electrically neutral. It has no direct immediate impact on intracellular or extracellular strong ion difference. We make almost 9 millimoles of carbon dioxide per minute at rest, and that means that we are generating about 8 millimoles of hydrogen ion equivalents intracellularly. And unless something sweeps up this hydrogen and associated carbon dioxide, we would literally melt. That is where the most important buffer in the body comes in, hemoglobin. This section covers material that I've already discussed in the course, but I'm going to take it a little bit further than the standard textbook approach. Most of the CO2 that is released from the tissues goes into red blood cells, where it dissociates into hydrogen ions and bicarbonate. Hydrogen ions bind to hemoglobin, causing release of oxygen, known as the Bohr effect. The deoxyhemoglobin then binds to carbon dioxide to form carbaminohemoglobin, the Haldane effect. Almost unnoticed but really important in the background is the shift of some of this bicarbonate into extracellular fluid in exchange for chloride to maintain electrochemical balance. This is known as the hamburger effect. But there is more. Chloride itself binds to hemoglobin, competing with 2,3-BPG and carbon dioxide for binding sites, and enhances the liberation of oxygen. This is a simplified diagram of hemoglobin that consists of 4 globin subunits, 2 alpha and 2 beta. Embedded in these is the heme group, and they coalesce into a central cavity. That's right in the middle. CO2 binds to the N-terminal amino groups of the alpha and beta chains. Chloride and 2,3-BPG bind to the positively charged central cavity. Regardless of the binding site, carbon dioxide binding to the N-terminal, hydrogen ions binding to the proximal histidine, and 2,3-BPG and chloride binding to the central cavity, all of these have the same impact to stabilize hemoglobin to the T or tense state, liberating oxygen. The histidine retains a positive charge, so that when CO2 accumulates, there is always electrochemical balance between the chloride, the bicarbonate, and hemoglobin. At baseline, always, the bicarbonate concentration of extracellular fluid is 24 millimoles per liter. That bicarbonate is kept steady by a number of mechanisms within the kidney that reabsorb and regenerate the solute. The mitochondria generate at least 200 mils per minute of CO2, or 9 millimoles per minute, and about 90% of this is converted to carbonic acid. CO2 travels to the brain, and in the CSF it is converted to carbonic acid as well. That is because there is carbonic anhydrase in the choroid plexus. So bicarbonate and hydrogen ions are released. The pH of the CSF controls respiration in the medulla. Alveolar ventilation increases if the pH falls, and vice versa. Eventually, all of that CO2 that is associated with bicarbonate is eliminated as CO2 gas by the lungs. The baseline bicarbonate remains unchanged. So although all particles of bicarbonate are the same, I want you to think of bicarbonate in two ways. The resting bicarbonate that is generally unchanged, and the bicarbonate that is associated with fresh CO2 production, all of which is eliminated eventually by the lungs. Nearly all the carbonic anhydrase in the body exists within red blood cells. As I just mentioned, there are some carbonic anhydrase in the choroid plexus adjacent to the ventrolateral medulla, the location of the chemoreceptors, and in the parenchymal cells, including astrocytes. But overall, there is very little carbonic anhydrase in plasma, extracellular fluid, and cerebrospinal fluid. And where there is conversion of carbon dioxide to bicarbonate and hydrogen ions in extracellular fluid, it occurs slowly, and then rapidly it's reconstituted to carbon dioxide. As a consequence of this, nearly all of the buffering of carbon dioxide, carbonic acid occurs within red blood cells associated with the Haldane and Bohr effects. A minuscule amount occurs in extracellular fluid associated with albumin, globulin, and phosphate. Peripherally generated carbonic acid is rapidly reconstituted. Reconstitution of CO2 from hydrogen ions and bicarbonate is not buffering. This is known as the Chatelier principle, and it represents what happens in a circular fashion with carbon dioxide. Carbon dioxide cannot buffer itself. So most of the carbonic acid is produced in red blood cells where it is buffered, and some bicarbonate is released to buffer metabolic acids in the plasma. CO2 that does not enter the red blood cell mostly remains dissolved in extracellular fluid or is converted to carbonic acid. But as it degenerates to hydrogen ions and bicarbonate, nearly as soon as that takes place, it reconstitutes back into carbon dioxide. As a consequence, despite 9 millimoles of carbon dioxide being produced per minute, there is minimal impact on pH, and even if hypoventilation takes place, the increase in hydrogen ion activity is minuscule in nanomolar concentrations, and only a mild acidosis occurs. I want to now move on and discuss the interaction between bicarbonate and chloride. Bicarbonate and chloride are both anions. They both contribute to electrical neutrality. But chloride is a strong ion, and it is part of the strong ion difference. Bicarbonate is a weak acid with a pKa of 6.1. Bicarbonate acts as an extracellular buffer, but intracellularly it is a component of carbonic acid. The Hamburger shift facilitates intracellular electrical neutrality, but there is an impact on intracellular and extracellular pH. This table was published by Weston and Prang in 2003 as a consequence of careful experiments, and it demonstrates that intracellular chloride is much higher in red blood cells versus other cells, nearly 70% of plasma concentrations. But compared with plasma, the strong ion difference is significantly greater within the erythrocyte, demonstrating the enormous buffering capacity and increased tolerance of hydrogen ions within the red blood cell. When chloride enters the red blood cell, it reduces the strong ion difference and consequently the pH within the cell. But the impact of this is eliminated by the binding of chloride to the hemoglobin molecule. The reduction in plasma chloride increases strong ion difference. It's alkalinizing as a result of the hamburger shift, and that curtails the reduction in pH in venous blood. Bicarbonate that is pumped out of the red blood cell replenishes bicarbonate that has been consumed, buffering metabolically derived organic acids. Let's look how this works in different respiratory scenarios. Here we have our standard situation where carbon dioxide is produced by the tissues and buffered by hemoglobin associated with the chloride shift. This results in the pH of venous plasma being 7.36. Now, if we administer anesthesia and wait for the patient to breathe spontaneously, they usually hypoventilate, sometimes quite significantly, and carbon dioxide builds up in both arterial and venous blood because of failure to eliminate. Remember, carbon dioxide production hasn't changed. So in this scenario, the pCO2 has increased, and there is an increase in CO2 uptake by erythrocytes and increased plasma CO2. All of this increases bicarbonate production and chloride uptake by red blood cells. The net result is an increase in the PaCO2 and a small increase in plasma bicarbonate and a small decrease in plasma chloride and increase in the strong ion difference. As time goes on, the magnitude of these effects increases, and this is what occurs in respiratory acidosis. You may then panic and put the patient on a ventilator, for example, with a respiratory rate of 30 breaths per minute. As I showed you in the previous tutorial, CO2 clears very rapidly. We are hyperventilating our patient, and CO2 production remains normal, but CO2 release from the lungs is increased. This enhances available binding sites for CO2 and hydrogen ions within the red blood cell. As a consequence, the plasma bicarbonate falls and the plasma chloride increases slightly. The result is a reduction in the PaCO2, a reduction in the plasma bicarbonate, an increase in the plasma chloride, and a reduction in the strong ion difference. This is characteristic of respiratory alkalosis. Now, let's review this tutorial. This was part 1 of a series on carbon dioxide and acid-base. I started with a discussion of the absolute basics of acid-base and discussed strong ions, strong ion difference, and carbon dioxide, and why the hydrogen ion activity is an unbelievably small number but clinically very relevant. I explained why chloride is important, how hemoglobin buffers carbon dioxide, bicarbonate and chloride, and hyperventilation and hypoventilation. In part 2, we're going to move on to the core part of this series. We're going to discuss acute respiratory acidosis, chronic respiratory acidosis, and acute respiratory alkalosis. Join me then, and I guarantee you'll learn something. So that's the basics of acid-base. Next time we're going to look at respiratory acidosis and alkalosis. If you're enjoying the tutorials, please subscribe.